Good day, everyone. Thank you for joining us today for our live webinar, which will be presented by our guest speaker, Xueli Guan, and entitled The ABC Approach to Explore a Fascinating World of Bacterial Lipids. My name is Maria Fedorova, and I'm based in the Technical University of Dresden and will be your host today. Like usual, this webinar will be recorded and will be uploaded to our Lipid Maps website and our YouTube channel within the next 48 hours. The webinar will last around 45 minutes and we will have around 10 minutes afterwards for the questions. So now I would like to introduce Xueli to you. So she is the head of the metabolomics platform of the Novo Nordisk Foundation Center for Basic Metabolic Research in Copenhagen, Denmark. Her work focuses on advancing and promoting metabolomics and lipidomics technologies to support and empower the broader metabolomic research community. She earned her PhD from the National University of Singapore. In 2011, she received the prestigious Ambizione Career Grant from the Swiss National Science Foundation and became a junior research group leader of lipidomics and systems biology at the Swiss Tropical and Public Health Institute. In 2015, Xueli returned to Singapore as assistant professor, and her research focused on developing novel strategy to characterize microbial lipidome and to understand the variations and functions of microbial lipids in relation to the human health and antimicrobial resistance. Xueli also plays an active role in the scientific community as a part of the International Lipidomic Society and its microbial lipids interest group, and she serves as a member of the steering committee to promote collaboration and standardization in this rapidly expanding field. So Xueli, the floor is yours, so please unmute yourself and share your slides, and we are looking forward for your talk.
Thank you, Maria, for the kind introduction, and a good day to everyone from probably different parts of the world. I'm Xueli, and it's my pleasure to be here today at this Lipid Maps webinar to share with you my work. Actually, I have to have a disclaimer. This is the work from my previous institution, and the focus is really to introduce to you the ABC approach to explore the fascinating world of bacterial lipids. So I don't want to undermine our efforts of elucidating lipid structure. We know it's not easy. So the take-home message is it's really not easy, but hopefully with more efforts and all this, we'll make it easier. And ABC approach here actually means analytical chemistry, biology, and computation. Yeah, and as Maria has pointed out as well, I'm currently based in Denmark. So the outline of my talk can be categorized into 4 M's, and the first M is to give a general introduction on microbes, particularly on bacteria, and why we care about lipids in bacteria. And the second part, after the introduction, I'll be sharing 2 of the studies that we had. The first study is on the development of Lipid ADA, a semi-automated computational workflow in combination with mass spectrometry that helps to elucidate the structure of the highly complex lipid A found in gram-negative bacteria with relevance to antimicrobial resistance. And the second story I'll be sharing with you is the discovery of 2 novel phospholipids in Escherichia coli. And this was enabled by the integration of metabolic strategy, genetics, and mass spectrometry. Finally, I'll just give a short overview of what we are doing in ILS with the microbial lipids interest group. So why do we care about microbes? Basically, we live in a microbial world, so besides our cells and organs, we have to coexist with lots of microbes in our body, which can be beneficial and also potentially harmful. And increasingly, it's known that the microbiome composition in our body varies and it affects health or disease status to some extent. More commonly known is the effects of pathogens that are circulating in the world, particularly with bacterial infections. There's a growing concern because despite the discovery of antibiotics, bacteria have become smarter and been able to overcome the actions of antimicrobials, and this is a statistics projection that antimicrobial resistant infection will be one of the leading causes of death in the future. So why is bacterial lipids so important then in the context of human health? So this is a cartoon of the membrane of gram-negative bacteria, which is highly complex. As you can see here, it has two bilayers which comprise of which is the outer membrane and the inner membrane, and in between is the peptidoglycan. With such a highly complex structure, it forms a permeability membrane, which makes drug discovery for bacterial infection extremely difficult because it keeps things out, including toxins, but also antimicrobials. The variations in lipid structure between microbes and human actually is a target for identification by host cells, and it is well known that lipopolysaccharide found in the outer membrane of gram-negative bacteria is able to trigger toll-like receptors in macrophages, and it is one of the mechanisms of sepsis. Given the differences in lipid repertoire of bacteria from the host, it also makes targeting bacterial lipids a potential target for drug development, be it at the enzymatic level or at the level of interactions with the bacterial lipids. And one example I like to highlight is LPS, lipopolysaccharide, which due to the lipid A composition, which has phosphate group, it is negatively charged, and cationic antimicrobial peptide has been developed as one of the potential last-line drug for treating gram-negative bacterial infections. And one example of this last-line drug is polymyxin, which will be brought up later on in my talk. We increasingly also learn that bacteria can secrete lipids, including signaling lipids like the very popular bile acids, short-chain fatty acids, as well as lipoamino acids, which are involved in cell-cell interactions, not just between microbes, but between the microbes and the host. Over the last few years, with the expansion of microbiome research, the number of bacteria that has been identified or are of interest have grown, and associated with it is the growth of the number of lipids and the type of lipids that have been identified, and novel new functions have also been reported. One such example is the gut microbiome bacteria Akkermansia mucinifila, which in 2022, the group of John Clady has reported that a branch-chain phosphatidylethanol from this bacteria is able to induce immunoregulatory functions. In addition, the group of Ruth Leith and others have found novel sphingolipids in the gut bacteria, which is able to, for instance, in this study, they have shown that the sphingolipid produced by the bacteria is able to enter the host metabolic pathway and impact host ceramide levels. So we are in exciting times with new discovery of new lipids in many bacterial species. And how do we approach this diversity then when there is both the components of the organisms itself as well as the lipid? So there are really many gaps in microbial lipidomics, and while we are very excited about studying lipids, we do have a bottleneck in this field because of three different reasons that I've categorized. The first is microbial diversity. It has been predicted that there are about 1 trillion species of bacteria that can potentially exist, and of which 10 million catalog species. And if we go down the line, 1,400 has also been reported to be pathogenic species. So in human lipidomics research, it's just one organism, and it took us many years to get to where we are today. So imagine this big number of microbes. How are we going to tackle so many bacteria or microbial species in terms of deciphering their lipid composition? So in our lab, we took a more targeted approach where we look at single species because of the hypothesis and the interest that we have, which is on antimicrobial resistance. And we have developed fragmentation-based rules based on the deep lipid profilings and annotation that we are doing. A possibility to screen more diverse bacterial and also from the microbiome context is to potentially use a molecular network spectrum matching, and some of the great works to share would include work from Makoto Arita's group in Japan, as well as Peter Dorestein in UCSD, where he actually reached out to the community and have curated MS spectra for more than 60,000 microbial monoculture, which we now can try to map tandem mass spec spectra to potentially identify lipids or microbial metabolites from this very comprehensive resource. The other gaps that we have in microbial lipidomics is the lack of lipid standards and databases, because bacteria and microbes really make a lot of weird lipids that are underreported, and because we are lagging behind in the field, there's still not many synthetic standards that are available, for instance, for LPS, lipoteichoic acids. In addition, the final gap I'd like to share is the variations between labs and a lack of harmonization. For human plasma, at least, there's the NIST SRM 1950 that many of us are using when we are doing human lipidomics work with plasma, but it's not the case for bacterial. And if we were just to take a look at E. coli, which is the classical model organism, and going through the literature, and we actually find many different reports of the types of lipids and the composition that based on the different studies. And you can see that in the 4 papers that we have curated here on the y-axis, and on the x-axis is the number of counts of, and the different color codings are the different lipid classes which are abbreviated. And excuse the abbreviation, but the take-home message is that there's different colors appearing in the different reports, as well as the length of the banding is very different. So all of us are reporting things very differently. But it's not about right and wrong in this situation. The situation is that it's just complex, because for bacterial lipidomics, we are growing them differently and we're using different strains. So in this context, the 4 different studies have been growing the bacterial in different media depending on their hypothesis. They used different strains and they collected the cells at different growth phases. And we know that different growth phases will have different lipid signatures. So in other words, what is then the true lipidome that we can see from even a single model organism? There's still no answer at this moment because of a lack of harmonization. But we believe we can build this together as a community. So in my lab, we have a very classical workflow and for bacterial lipidomics and metabolomics, because we can actually analyze the lipids and metabolites from the same extract, same bacterial pellets. And as mentioned in my introduction, the ABC literally means analytical chemistry, biology, and computation. So we typically culture the bacterial in the lab, and one source of the bacteria can also come from our clinical partners. And after sampling, the routine that we have is to perform liquid-liquid extraction using chloroform methanol. This is then analyzed using liquid chromatography and high-resolution mass spec with automated MS. And we perform data analysis, pre-processing, and identification using both public databases as well as in-house theoretical and experimental database. As mentioned, from the same sample, we can have multiple fractions and not just fractions for metabolomics from the upper phase of the liquid-liquid extraction, but for gram-negative bacteria from the same extracts using Blay and Dyer or Foch extraction, where we have the phase partitioning, we can isolate the phospholipids from the lower organic phase, and for lipid A, we can get it from the intermediate phase, and by acid hydrolysis to cleave off the sugar component, we retain the lipid A component from the bacteria, which can be subjected to LC-MS analysis for subsequent studies. So the first example that I would like to share with you is Lipid ADA, which is the semi-automated workflow to elucidate the lipid A structures that enable us to help probe the lipid A repertoire of antimicrobial resistant bacteria. To give a bit of more introduction about lipid A, it is the endotoxic component of gram-negative bacteria, very well known in its involvement in virulence, as well as antimicrobial resistance. So lipid A is the lipid anchor for lipopolysaccharide, and it is the known potent factor for triggering immune responses and is involved in sepsis. So the well-known lipid A isolated from E. coli, the major form, is this diphosphorylated lipid A, which has 6 tails, so it's hexa-acylated, and it's known to be a strong toll-like receptor for agonist. Its precursor lipid 4A, which has only 4 tails, is on the other hand a toll-like receptor for antagonist. With the known immunomodulatory effects of lipid A, modifications has been made and monophosphoryl A has been commercially available as it can serve as a vaccine adjuvant. So besides the immunomodulatory effects that can be altered with changes in lipid A structures, other functions can also, and in terms of the bacterial physiology, can also be influenced by lipid A structural modification, as can be shown here. So lipid A has this 2-phosphate group that are negatively charged, and this negative charge actually are involved in their interactions with cationic peptides, which is one of the drugs that I mentioned that can be used to treat gram-negative bacterial infections. Under environmental cues, which can activate the 2-component sensor kinase PMRAB, there is the possibility to modify lipid A, for instance, with phosphoethanolamine hit group. And this group actually reduces the negative charge of lipid A. And when the negative charge is reduced, this reduces the interaction with the cationic antimicrobial peptides. And modification of lipid A is indeed one of the known mechanisms for polymyxin resistance. On the other hand, bacterial can also add another phosphate group to the diphosphorylated lipid A to make a triphosphorylated form. And here, this triphosphorylated form has been associated with dioxycholate resistance, and this is one of the possible mechanisms why gram-negative bacteria can survive the harsh conditions in the gut. So these structures are not here to scare the audience, but we can see that the structures are highly complex, and it's not trivial to solve lipid structures then. But we have good instrumentations, and this allows us to resolve the structures at high resolution. And this is a cartoon of the tandem mass spectrum of lipid A isolated from E. coli. And as you can see, they form quite a systematic pattern. So we ask ourselves, can we automate the process can we learn from this pattern and automate the process of elucidating lipid A structure? So what we did was both experimental and theoretical. We grew E. coli and Pseudomonas aeruginosa in the lab, and the reasons for choosing these two bacteria as our reference is because their lipid A biochemistry is very well established, with the lipid A structures elucidated, and the major enzymatic machineries are also very well established. So we extracted the lipid A from this bacterial and performed LCMS analysis using different collision energies. And we then curated the fragmentation profiles and came up with fragmentation rules. This is further supplemented through rigorous literature review, owing to the many work by others which have performed lipid A analysis and very high quality fragmentation that has been made available publicly. So with this combined, we came up with the fragmentation rules and a theoretical database of possible fatty acids, hate group, and cross-ring structures that can be formed during defragmentation, and coded into a Python script workflow that we named LipidAder. So the output of LipidAder can produce the lipid A identification at 3 levels, with the base level just at the very fundamental level, which is just the lipid A and the type of modifications on the hate group. The second level, it's based on the sum composition of lipid A as well as the hate group, and the sum composition, it's the fatty acid carbons, the number of double bonds, and the number of hydroxylation. And the third level would give more details, and it includes the fatty A cell composition as well as position. As you can see here from this example, this lipid A has been resolved with the hate group modification as well as the fatty acid information, and the order is the position information. So with this training data set, we have built the workflow and generating this output. We then apply this workflow to look at the lipid A of the less characterized bacterial, and we have focused on the escape panel of pathogens, which I will bring up after this slide. So the output I'd like to share is also generated in the MSP format, because this will allow us then to integrate with other open source software such as MS-DAO or MetaboKit, that we can then match to this library and perform potentially semi-quantification with the peak areas. So we applied Lipid Ada to probe the landscape of Lipid A in this group of pathogens known as ESCAPE, and they are highly relevant because they are pathogenic, and WHO has categorized them as priority pathogens because of their emergence of resistance. And the gram-negative members of ESCAPEs include Enterobacter, Klebsiella pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. In addition, we also put in a relative of Klebsiella pneumoniae, K. aeruginus. And E. coli always remains to be our reference model. So this is the fingerprint of the output of Lipid Ada when applied to these 6 different species of bacteria, and some of which we have also different strains. And the y-axis is the mass to charge ratio, and the x-axis is the retention time. And the different color codes and shapes refers to, in terms of the color code, it refers to the type of hit group. For instance, monophosphorylation has a light gray, and ethanoamine hit groups have different shades of blues. And the degree of acylation is represented by the different shapes. And if the cross is 2 acyl chains and up to 7, which has a round shape. This map highlights the diversity of lipid A that we can that exists in this bacterial, and in total we have been able to solve the structures of 188 unique lipid A from this panel of bacterial. With this work, we are also very thankful that subsequently, and actually very recently, lipid maps have a new partial spectral database, and the lipid A structures that we have resolved using Lipid Ada is now available with on Lipid A, and thanks to the work of me, Laura, Paul, Miguel, Maria, and colleagues from Lipid Maps and EpiLipidNet. So what do we do with these structures? From our perspective, with the interest of the group, we want to use it further for biological studies, and one of the utilities of this Lipid A atlas that we have built is that we can actually differentiate bacterial species and strains using this biochemical, high-resolution biochemical phenotype. So this is a heat map representation of the output that we have seen. And in this case, we group the Lipid A into 3 blocks. The first block is the heat group modification. The second block is the fatty acyl compositions in terms of the numbers and where the positions are. And the third block is the chain length of the different fatty acids. And in this particular heat map, it's the normalized count of each type of lipid A based on their chemistry. So we can basically cluster the bacterial species and strain based on this lipid A signature, which, as you can see from the different color banding, they are very, very different. And so the first example is species discrimination. So Pseudomonas aeruginosa cluster very far away from the other species, which is not unexpected because its lipid A tend to have an enrichment of 5 tails, for instance. And the other example that I'd like to bring up is boxed in green. So Enterobacter homaceae, it's actually sequenced by a colleague in my former institution, and it is a close relative of the other Enterobacteriaceae. So it does make sense that the Enterobacter cluster more closely together. Next, I'd like to bring up how this can be utilized for strain discrimination, and in collaboration with clinical partners in the Singapore General Hospital, we managed to get clinical isolates from patients. This particular patient, unfortunately, was infected with Acinetobacter baumannii. Originally, the strains that was isolated is polymyxin sensitive. Subsequently, the clinicians isolated from the same patient resistant Acinetobacter baumannii. So if you recall, resistance to polymyxin, one of the mechanisms is this phosphoethanoamine capping. So this pair of isolate actually serve for us as a positive control. And if we look into the heat map at the position of pre-1 and post-1, we do see that there's accumulation of heat group modifications. What we also found interesting is that in the other strains that do not have any known resistance to polymyxins, there was also accumulation of the capping in the case of this E. coli strain EC958 as well as Klebsiella, which has the error 4 and modification. So we ask ourselves, is modification with this phosphoethanoamine heat group or this error 4 and heat group the sole reason for or mechanisms for resistance? So we went back to look into the data generated by the SGH colleagues because they have earlier published the genome sequence of these strains, and one of the mutation is PMRB, the two-component sensor kinase. And this kinase is involved in the regulation of a few enzymes, one of which, of course, is involved in the addition of phosphatidylethylamine to the LPS core. But another modification that we took note of is acylation, deacylation. So with this atlas that we have generated with lipid ADA, we could then use it in combination with lipid blast to extract the peak areas to look at the relative intensities of the different lipid A's that we have identified. And this is a Pablo plot of the relative ratios of the different heat groups of lipid A found in this two strain with the fold change on the x-axis with the resistant strain calculated over the sensitive strains. So you can see, as expected, the phosphatidylethylamine heat group enrichment, it's found here, so it's consistent with what we found. And in addition, we also found that there is a significant increase in the degree of acylation for this resistant strain in contrast to the sensitive strain. In a nutshell, it's not just the presence of phosphatidylethylamine that is the signature of this resistant strain, but it's a combination of multiple modifications that are found in this resistant strain that can then be potentially be associated with the resistance profile. So in summary, for this part of the talk, I hope I have impressed upon you that the fine details of the lipid structures, the combination of lipid chemistry and the quantity matters to the functions, and that natural variation in bacterial lipids has a strong link with antimicrobial resistance phenotypes. So I'll move on to the next study that we have is where we have revisited the lipidome of Escherichia coli, which is a friend as well as an enemy in human health. So E. coli is not just a model organisms that we use for our research or in synthetic biology, but it can be a pathogen. It is also a resident of our gut, and in fact, there are pills out there with the Nysol 1917 strain that is commercially available. So that's where the friendly part is. So this study is actually arising from an incidental finding in our works where we were attempting to catalog the lipids of the gram-negative panel of escape pathogens. And what we found consistently during our analysis of phospholipids in our work is that in E. coli, there's always this peak that elutes earlier than the PS standard, but we are very sure it's not due to retention time shift arising from a bad chromatography. And when we zoom into the tender mass spectrum of this particular peak, we found that it has a neutral loss of 101. In comparison, for phosphatidylserine, the classical signature of its tender mass spectrum is the neutral loss of 87, which comes from a serine. So between 101 and 87, it's a mass difference of 14 Dalton. And in lipids, it's very common to have 14 Dalton that arise from a CH2, which means a chain length difference of 1 carbon. So we ask ourselves, what can be this potential new signature? We also ask ourselves whether this signature is unique to E. coli, and we have been cataloging the lipids in a very systematic way for the gram-negative members of escape pathogens. And so we mine our data, and look into the same set of strains that you have seen in the Lipidator story. So interestingly, we find that this unknown peak is consistently found in all Enterobacteriaceae species, but not found in the other gram-negative species like Pseudomonas or Acinetobacter baumannii. In addition, we also put in another control, which is a gram-positive bacteria, Staphylococcus aureus, which doesn't make PE or PS. So the unknown is not found. It took upon us, like our technology have improved and allowed us to detect a great diversity of different lipids, and now we actually are in a position whereby we have a lot of unknown peaks. How do we approach it? The way to approach it for this study is to use metabolic strategy, and for that we would like to first reflect upon it, because we also need to choose which type of metabolites or which kind of strategy we need to use. So we took one step back and think about what can be the potential source of production of this endogenous lipid. And there are three possibilities that we explored. The first is an uncharacterized enzyme, and it initially was very promising because a Dalton 14 means it could potentially be threonine, and threonine is a major amino acid, similarly to serine. And in the literature, phosphatidylthreonine has been reported in parasites, and more recently it has become a very interesting molecule also from human health perspective, with the discovery of phosphatidylthreonine in human blood, which is associated with coronary artery disease. So that would be our first candidate. But when we tried to blast the E. coli genome against the parasite genome, we were unable to find any similarity between for the phosphatidylthreonine synthase enzymes. The second possibility is non-enzymatic reaction, for instance, with the transfer of a methylation transfer, but this is very unlikely because it is a mutagenic reaction, and we did not subject our bacteria to strong mutagens, and those are done in very routine cultures. So the last possibility is, could it be formed from a known enzyme but with unknown function? And one of our candidate pathway is phosphatidylserine pathways, and phosphatidylserine is a precursor of forming the major lipid in E. coli PE. So what we did is to fit the bacteria under minimal media conditions of potential substrate that we postulated that has a Dalton 14 difference from serine that are available commercially. And we subject then the fed bacteria to lipid extraction and LC-MS analysis. So here is a recap of the different peaks that we detected in E. coli, which is eluting earlier than the PS standard. And in Pseudomonas, we have reported that PS exists, but we did not report this potentially novel lipids. So the candidate substrate that we could get hold of commercially are threonine, which is this natural amino acid, allothreonine, a rare isomers, methylserine for methylation of to represent methylation of serine, and homoserine, another natural amino acid. So with this, we found that Pseudomonas, but not E. coli, was able to incorporate threonine, but this phosphatidylthreonine elutes at a time that's in between the E. coli lipid and PS. So this rules out the possibility that threonine is the substrate. For allothreonine, the bacteria was also able to incorporate it into its lipids, but it has a retention time that is close to PS, but not the E. coli peak. So it rules out the possibility of allothreonine. Methylserine elutes very early again, then this rules out the possibility. And finally, we have homoserine, which elutes very closely to the E. coli peak. So from this metabolic strategy incorporation work, we have confirmed that this E. coli endogenous lipid is phosphatidylhomoserine. So the next question that we ask is, how is this lipid formed? So as mentioned, the candidate pathway is PS synthase. We then contacted our collaborator, Cheng-Shu Sin from NUS, who has the mutant for PS synthase, where my hypothesis would be that if this lipid is formed through this pathway, we will not be seeing that peak in the mutant. In addition, if we were to feed the mutant with homoserine, we will not see the accumulation of the peak. So what we did was to grow wild-type E. coli and the mutant for phosphatidylserine synthase at baseline condition and also fed the cells with homoserine. So this first set of plots show the usual E. coli under wild-type condition where there's the accumulation of this peak with the major the XIC of the major species for phosphatidylhomoserine. And in the PSSA mutant, we totally lost the signal. This helped us to confirm that this endogenous lipid is formed through the PSSA pathway. With the incorporation, it further confirms that there is no accumulation of phosphatidylhomoserine in the mutant. And this definitely helped us to confirm that this lack of accumulation is not due to limited substrate, as we have gave quite a high concentration of homoserine. To further confirm the enzymatic profile of PSSA and its ability to use homoserine as a substrate, we collaborated with Jong-wook Kim in Korea, and which he published the structure of PSSA of E. coli just last year. So his PhD student Yunju performed the analysis of enzyme kinetics where homoserine is used as a substrate with PSSA isolated from E. coli, and they have helped us to confirm that PSSA indeed can use homoserine as a substrate, a bit at lower affinity. During our study, with the incorporation of homoserine in E. coli, we actually also found another peak that is accumulating and eluting slightly behind phosphatidylethylamine, as can be seen from this chromatogram with the red arrow at this little bump. So phosphatidylethylamine has this classical fragmentation of 140, which is representing the phosphoethylamine hate group. And phosphatidylethylamine is formed from PS through the action of PSD. We also performed tandem mass spec for this particular peak, and what we found is that this peak has a signature of 154, which is a Dalton 14 difference from the hate group of PE, and we postulate that this is highly likely to be a propanoamine hate group that is arising from the decarboxylation of PHS to form phosphatidylpropanoamine. I'm not showing it here in the slides, but if you refer to the published manuscript, we actually retrospectively mined the data of the other bacterial species as well as E. coli and confirmed that PPA can also be found endogenously in the bacteria. Finally, we look at how phospholipids remodel when the bacterial is treated with homoserine. And the key finding that we have is that when the bacteria is treated with homoserine, it accumulates phosphatidylhomoserine as well as PPA. But at the same time, we found a strong downregulation of PE and PG. And this can be explained through the pathway as they share the common intermediate CDP-DAG. So when the cells are given homoserine, it channels the substrate to PHS and it compromises the formation of P and PG. So this study overall allowed us to look at the remodeling of E. coli and has helped us to revisit the biochemistry of E. coli through the combination of metabolic incorporation, LCMS, with tandem mass spec and genetics. We have shown that E. coli can make PHS through the PSSA pathway, and PHS can be decarboxylated to form PPA. We have also demonstrated the substrate promiscuity of known enzymes, and this led us to stronger reflection. Bacteria are not naturally growing in the lab, which we just give them synthetic media, but they are actually exposed to the wild. So the wild has many more substrates that the bacteria is exposed to, and so the question is, what is then the true lipidome of the bacteria when they are exposed to the natural environment instead of the synthetic environment? And many questions has to be answered in the future, which we don't have now. So with this very fascinating world of bacterial and lipids, we tried to get together as a community and have established the interest group at ILS. And basically, the problem statement is that there's really a huge diversity of lipids, but there's a lack of standardization, and microbial lipids are so important in human health and disease. So the vision is to contribute towards community level efforts and establish standards for microbial lipids. And one of the key activities that we have is the inter-lab study that we are trying to initiate, where we hope to define consensus lipidome and provide recommendations for microbial lipidomics. So this exercise already started 2 years ago in 2023, where the steering committee members, including myself, Jenny, Kelly, and Justin, actually tested extract, and we have to also acknowledge MetaboHub and Justine for doing the production. And with this testing being sorted out right now, we are in the implementation phase where we have reached out to participants, and we are in the process of rolling out the materials distribution and bioanalysis. Although we do not have the data, but we do have some things that we like to share in this community, also from an education perspective. Because during the testing phase, we actually found, of course, the classical lipids with the different methods as can be shown here that we populate on top of the 4 literature that I have shown earlier. But in addition, we actually found 3 different classes of lipids, and they are phosphatidyl-methanol, and PA with modification with methyl carbonate and succinyl modifications. And I'd like to share with the community because these are actually artifacts that are generated. And I strongly also recommend this paper from the group of Guan Ziqiang in Duke in US, where they have already published such artifacts in bacterial lipids. And it is underreported partly because PE is major lipid in gram-negative bacteria, but in mammalian cells, it's PC. So we don't see this kind of modification as commonly in human lipidomic study. So one needs to be extremely careful when dealing with microbial lipidomics. So I'd like to give my concluding remarks that the precise structural information of lipids really codes for functions and help us also to elucidate lipid metabolic pathways. This field is really very exciting, but with so many lipids and so many microbes, the road ahead of us is really long, but we look forward to it. And with that, I'd like to acknowledge my former team in NTU, my collaborators from Singapore who contributed to the work, as well as from Korea. And these are the funding sources that had enabled these works to be done. Thank you for your attention, and I'm happy to address any questions you may have.
Thank you very much for this really great presentation and this overview of complexity of bacterial lipids, which is absolutely fascinating. So we have some time for the questions. We have one question in the Q&A which is very practical, so I will just read it out. And Hussein here is asking which software you have used for the heat map generation.
Oh, for the heat map generation, it's either R code or a Python code that we script ourselves.
Thank you. We have a question from Susan, please.
Hi, how are you? Thanks very much, Xueli. That was absolutely fantastic to hear about your work and to see it in such fantastic detail. And I love the approach of the combined approach of genetics with mass spectrometry on a strain specific level, and it's very applicable. So basically, I suppose my question is the major lipid producers in the gut microbiome are generally the Bacteroideid species. There's a lot of information in terms of the range of lipids and the diversity of lipids, and some of the pathways that are involved there. Some of that work has been done again by Ruth Lay, but also by David Clark here in Ireland. And I just wondered, have you ventured into looking at Bacteroides in similar detail and in terms of their interactions and their potential?
The quick answer is no.
Okay.
We haven't dealt with the bacteroids. We actually dealt with Akkermansia and a few other gut microbiome bacteria, but not bacteroids. Yeah.
Oh, okay. And to follow up on that, I just suppose that to ask if you know, I've always considered the microbiome as in its initial stages as chock-a-block full of E. coli strains, which are commensal strains. So have you considered commensalism in this picture? Clearly, the role of pathogens is important too, but what about training of the immune system and commensalism? Is this something you guys are considering here as well?
For us, not at the moment, because originally our research was started with the drug resistance mechanisms, but progressively we realized that we should not be so restricted, and because a lot of bacteria actually now we know makes very similar lipids to make lipids that are very similar to humans, just that there is modifications. For instance, the sphingolipid structure is very different between the bacteroids and the humans. So it is a big opportunity now to expand to the many other bacterial species. And for the microbiome space, the number of bacteria is really enormous. And that is also why the motivation to get the community together, because all of us are publishing one lipid and one bacteria one at a time. I think it's important to consolidate everything and have databases where lipid maps is actually one of the central.
Thank you. I really enjoyed your presentation, and thank you very much for that and for answering my questions.
Thank you.
Thank you, Susan. So I will ask my question. So you actually commented on that, and this is just like a general maybe question. I don't think we all have an answer, but you already mentioned that, okay, the variability of bacterial lipids, like with lipid A examples, it's absolutely fascinating, right? And you showed that the different strains, the different conditions, everything is basically changing that, right? And at the end of your talk, you also postulated this kind of question problem, right? That we study bacteria lipidome basically in vitro by culturing them in certain conditions, and how does it actually reflect the in vivo situation? And since I'm not from the field, but is there any ways how you can simulate at least, I don't know, like gut microbiome conditions in which you can grow bacteria to address, let's say, the lipidome, maybe not in the wild, somewhere in the nature outside, but at least in this microbiome, human microbiome direction?
So it's a very good question, and what we did in the lab was to use artificial media, because for the project on E. coli, one of the former PhD students was challenged to look at E. coli adapted in so-called urine media versus gut media. And there are literature that has been published for this kind of synthetic media. And the reason for choosing these two media is because E. coli, it's known to be involved in the gut, but also in UTI infections. So, but no matter what, media are still very artificial, right? So, but at least those two media are better than growing it in LB or the minimal media, which biologists would like to use depending on why they do something. So there's a good answer, but I think also from the microbiome space, a lot of colleagues are using bioreactors where they can change the composition to mimic more the gut space. So I'm not a microbiome expert, and probably we have to talk to the microbiome experts themselves on what are the better setups to mimic the human body.
We have a question from Miguel.
Thank you very much for the talk. It was really great to see the lipid A part and also the discovery of these different lipids. Very interesting. I would like to ask whether if the data that has been observed related to the different lipid A modifications has been somehow studied in the context of the inhibitory concentrations of different polymyxins related to the different resistance of bacteria that or bacteria that have acquired resistance. So I don't know whether there is something semi-quantitative that has been studied and relates these modifications with the acquiring resistance.
There is, to my knowledge, there's no systematic analysis at the moment, because for antimicrobial resistance, what the clinician want is just a rapid readout whether it's resistant or not. So they tend not to titrate in their analysis the different levels of polymyxins to look at things, but they always have a window of what is resistant or susceptible. So to my knowledge, there's no such works available.
It's very interesting from the mechanistic point of view.
From mechanistic point of view, also not that I know of, but I know that the interest is more like how is the resistance arising, because it's also not very clear. Yeah.
Thank you very much.
Thank you for your question, Miguel. So maybe in the last minute we have, I will ask one more question I have. So you started, let's say you started lipid A for probably one of the most of the people in the world so far, right? Can you say how many modifications can happen to lipid A?
Actually, it's pretty conserved. It's not that many, although it can be many. Because if you look at the structure, there's two hit groups, the glucosamine hit group, then you see, oh, there's two hit groups that can be modified. But actually, there's not many types of modification. Like you don't find choline addition, although choline is potential substrate. You don't find inositol, like glycerol modification. But it is very bacteria specific as well. So the capping for A-BUM was phosphatidylethylamine, but in Klebsiella, the more common capping is the Ara4N. Yeah, so the greater diversity also comes from the fatty acid tails. They can make pretty strange fatty acid tails as well for bacteria, as you probably have also seen. Yeah.
And what about the sugar component? How much variability that brings in? Do you have any idea?
Oh, for the lipid A, the most popular one, it's the diglucosamine, but there are also other bacterial that doesn't use this as the substrate for the sugar core. Yeah, so there are other modification as well, and here I can add the disclaimer. So our fragmentation pathway is based on the common core with the glucosamine groups, but not the other rare cores for now. Yeah.
Yep. Thank you very much for this great talk, for this really fascinating work of bacterial lipids and this very elegant work you show at the end with actually identifying these new lipid classes. I think this is also super exciting field to find this new lipids which we still don't know about. And so I would like to thank you, Xueli, for the talk and everybody who attended the webinar, and wish you a good day. Bye-bye.
Thank you. Bye-bye.